This is the final part of Chapter 2, The Chemistry of Life. This is Part 2G, and we're going to be discussing enzymes. So the opening question here for Section 213 is, can chemical reactions be speeded up? So the first thing I want to tell you is that in your cells, there are just, I can't even tell you how many, thousands and thousands and thousands of different chemical reactions that are occurring. And for example, starches might eventually break down into sugars, but that is a chemical reaction that goes on during digestion. But the interesting thing is it goes much faster, at a much faster rate, when the protein amylase is present. And so amylase is found in saliva. You have salivary amylase. And so if you eat, for example, a cracker that has starch in it, and you chew it in your mouth, the saliva that contains the amylase can break down that starch much quicker than if the starch was just kind of not, did not have amylase present. So we'll be talking about this whole idea of enzymes. Enzymes are proteins that their job is to speed up chemical reactions in cells. So enzymes speed up chemical reactions, and I mentioned there's just so many different chemical reactions that occur in the cell. When you look at the sum total of all the chemical reactions in an organism, that is called metabolism. And an enzyme is a protein that speeds up a chemical reaction involved in metabolism that will speed up the chemical reaction without actually being changed itself. And pretty much almost every metabolic reaction that occurs in the cell occurs with the help of an enzyme. So they're very, very important. Now, how do enzymes actually speed up chemical reactions? Well, they do it by what's called lowering the activation energy. So the activation energy is the amount of energy you need to get from reactants to products. And you can kind of envision it like a high jumper. So maybe an Olympic high jumper, could jump over a very, very high bar, right? But it would take a lot of energy, and that energy is like the activation energy. Now, the job of enzymes is to actually lower the bar. For example, if you're using the analogy of a high jump, lower a bar so that more reactants can become products. So if you lower the bar, maybe you don't have to be an Olympic athlete to go over the high jump. Maybe you can just be, sort of an average athlete. And if it's really, really low, you could have a lot of people that could actually jump over it. And that's the job of an enzyme, is it lowers the activation energy to get from reactants to products. And that then allows the reaction to take place more quickly. Now, each enzyme is a particular shape, 3D shape, and that enzyme recognizes one specific target molecule, a reactant, that is called its substrate. So as an example, we have here the enzyme lactase. So that's here. So the lactase enzyme is an enzyme that allows people to break down the sugar lactose. Now we mentioned before, lactose is a disaccharide, So you can kind of see it's here. There's two monosaccharides linked together. And the lactase enzyme, remember, is a protein with a particular three-dimensional shape. And part of the lactase enzyme is the part where the substrate will bind. And that's kind of what I've outlined in red. So this is where the lactose will bind to the lactase enzyme. And the place where the substrate binds on the enzyme is called the active site. And this interaction between the active site and the substrate is very, very specific. A different enzyme will have a different active site, and it will bind a different substrate. Once the substrate binds to the enzyme at the active site, the enzyme and the substrate each change a bit to slightly, just change a little bit, slightly different, to allow for a nice tight fit. And then the next part of this is that the chemical reaction takes place where the substrate becomes products. And in this case, it's glucose and galactose, which are the two simple sugars, the two monosaccharides that make up lactose. And then once the chemical reaction has taken place, the products are made, this lactase enzyme can be reused. It's not used up in the chemical reaction. But the idea here is that this enzyme makes it possible for this particular chemical reaction to occur at a faster rate that is very helpful to the cell. Without enzymes, some chemical reactions occur so slowly that they pretty much barely happen at all. And so enzymes are absolutely essential for the function of the cell's metabolism. Now, there are inhibitors that are molecules that can actually prevent enzymes from working. And here you can see what's called a competitive inhibitor. And what a competitive inhibitor is it has a similar structure to the substrate, similar enough that it will bind to the active site of the enzyme. When it binds to the active site, which you can see over here, then that prevents the substrate from binding. When it prevents the substrate from binding, then the reactants, the substrates, cannot become products. So this is how a competitive inhibitor works. A competitive inhibitor mimics the shape of the substrate, binds to the active site, and then prevents the substrate from becoming product. There are also inhibitors called non-competitive inhibitors. Non-competitive inhibitors will bind to the enzyme, but they bind in an area that is not part of the active site. That's what you see here. Here's the active site. And you can see this inhibitor binds to a different spot. But when the inhibitor binds, it changes the shape of the active site so that now the substrate can no longer bind. And when the substrate can no longer bind, once again, it has inhibited the chemical reaction. Now, some drugs are actually work by inhibiting enzymes. And we do have some naturally occurring inhibitors found in cells that help to control enzyme activity. So remember, enzymes are a type of protein, and like all proteins, enzymes function depending on their shape. Changes in the shape can prevent the enzyme from functioning properly. Now, we mentioned lactase is an enzyme that breaks down lactose, which is a milk sugar, and there are some people that are lactose intolerant. Those people that are lactose intolerant, they actually have gene mutations in their DNA that change the amino acid sequence of the lactase enzyme, and that changes the shape of the lactase enzyme, making it ineffective, leading to lactose intolerance. So once again, this is an example of the shape of the protein is absolutely essential for its function. And if there is some type of change in the shape due to, for example, a genetic mutation, that enzyme can no longer be effective and do its job in a particular chemical reaction. So this is the end of the Chapter 2 lecture. Make sure that you read the chapter, that you write out the review questions that have been posted on Moodle, and study the material so you're ready for the exam. Also review the Mastering Biology questions after you have done the chapter assignments. And there are some extra material within Mastering Biology, practice quizzes, for example, note cards that, virtual note cards that can be very helpful for studying. So definitely check those out in the Mastering Biology website as well.